All right, I think we have a team on ready. So good morning, everybody. And again, Friday morning, 8:30, every week, the same thing, the wonderful science. And no exception today, we have fantastic scientists joining us and giving his thoughts about research. Dorota.
Hi, everybody. And first of all, thank you, Atul, for agreeing to talking to us. We are also interested in big data science. So I met Atul some 2-3 years ago when he was giving an amazing talk back at UCSD. And I just felt if we want to hear something interesting about big data and innovative way of using it, this is the person to hear. So without further ado, Josiah, please introduce formally. Thank you.
Good morning, ladies and gentlemen. My name is Josiah Toe. I'm one of the postdoctoral research fellows with Dr. Andrew Brown. Thank you for your time and attention. It's my pleasure and honor to introduce Dr. Atul Butte this morning as our guest speaker. Dr. Butte is a multidisciplinary, versatile visionary who wears multiple hats. He's a pediatric endocrinologist, a computer scientist, a computational institute director and principal investigator, a communicator and educator in academic and public forums such as TED Talks, New York Times, and Wall Street Journal. And to round it off, he's an entrepreneur and founder of 3 investor-backed companies utilizing big data, artificial intelligence, and medical genome sequencing. Dr. Butte completed the distinguished combined BA, MD in computer science and medicine from Brown University, and then went on to complete a medical residency in pediatrics with a fellowship in pediatric endocrinology at Children's Hospital, Boston. He then went on to complete a PhD in health sciences and technology at Massachusetts Institute of Technology and Harvard Medical School before becoming an associate professor of pediatrics and chief of the Division of Medicine, Systems Medicine at Stanford School of Medicine. Currently, he's the director of Bakar Computational Health Sciences at Institute, as well as the Priscilla Chan and Mark Zuckerber Distinguished Professor at UC San Francisco. Additionally, he's the chief data scientist for the entire UC health system with over 1,000 care sites. Throughout Dr. Butte's career, he has published over 270 peer-reviewed papers, is the owner of 24 patents, and has garnered a multitude of awards and recognitions. He was recognized as the White House Champion of Change in Open Sciences for his advocacy in utilizing publicly available data for research in 2015. He was also elected into the National Academy of Medicine. Dr. Butte has advocated on several occasions, and something I can deeply resonate with, is that if you want to change the world, you can't just keep writing papers. You have to do something and take technology out of the institution. Without further delay, please join me in welcoming Dr. Atul Butte.
Great. Well, thanks for that introduction. That's probably the best introduction I'll get all year. The remainder of my usual Zooms are people screaming at me why they're not getting enough COVID vaccine to distribute. So that's usually, this is much more pleasant Zoom than the average one in my day and week. So anyway, I got you for the next hour or so. So thanks for putting up with me. First, I'm obviously a medical doctor. You're all medical audience. I got to start with my disclosures. I have just a few. Okay, maybe I have more than just a few, but suffice it to say, I've started a bunch of companies on top left. I consult for a bunch of companies in the middle. I don't blame you if you don't believe another word I say over the next 45 minutes. I'm also proud of the list on the right. Those are all the companies started by my students and grad students and postdocs. They don't always include me, but they almost always include as a platform data. It's often big data and it's often open big data. And these are the companies that are started by students, even if they go into academia. So I'm going to show you how they do it. I'm going to show you how I do it, and maybe I'll convince you. It's the most amazing time to be in biomedical innovation and even entrepreneurship, even if we're in academia, which I think most of the audience here is. All right, so just to get started and level set a little bit, obviously big data, AI, machine learning are all over the place, certainly in magazine covers like these, whether it's Nature, Science, Fortune. In fact, The Economist covers it again and again, the importance of data. On the right there, the bottom right there, is a more recent cover from The Economist, which it's fascinating. Basically suggesting that data, they're showing oil rigs there in the water, and they're basically suggesting, as people have said, data is the new oil. Now, I hate that phrase because when you think about oil, you think about oil barrels. And if you know an oil barrel, either I have that oil barrel or you have that oil barrel. We can't really both have it. But data is different. I can have data and build something with that. and you could have the same data and build something different with that, right? So data is divisible and really scalable, infinitely scalable, and we don't even have to compete with our uses of data. So the phrase I like, which comes from yet another TED Talk, is data is the new soil where you plant ideas and watch them grow or help them grow. So I don't think it's next to oil, but certainly these are the phrases that are out there at the level set to make sure everyone gets them, because this is particularly important in ophthalmology, because these phrases are around. Here are just 3 of the more common phrases used. Artificial intelligence is when computers mimic aspects of human intelligence. So cars that drive themselves, languages that get translated, that's AI. It's just a general mimicking of human intelligence by computers. Machine learning is a big phrase that's used, where you're teaching the computer how to do AI by giving it data. Now you might think about that and say, that's a no-brainer. How else would you teach a computer except by giving it data? Well, for decades, the only thing we had to give to a computer were rules. We didn't have enough data, right? And so in the 70s and 80s, as we'll see in a moment, the big AI medicine revolution back then was to go ask doctors, let's say, what do you do when you see this? And what do you do when you see that? Gather up those rules and then teach the computer the rules. And obviously that doesn't work that well. In fact, what's obvious now is that it's much better to see in biomedicine what a physician does rather than ask them what they're doing, right? It's better to see what do they actually do, the data instead of the rules. So machine learning is teaching a computer AI by using data. In there, we have supervised and unsupervised. And so supervised means we have a right answer for this question, right? We know if it's diabetic retinopathy or not, right? We know if it's stage 4 breast cancer or not. So computer, why don't you see if you get that answer? We can measure sensitivity, specificity, precision, accuracy, right? We have a right answer. We're trying to gauge the computer to see if it can come up with the same right answer. Unsupervised means we have no clue what the right answer is. There is no answer here, right? We have a ton of data. We're trying to get it to organize, to understand it better. So that's where clustering, hierarchical clustering, k-means clustering, things like that come into unsupervised learning. And then finally, you have deep learning, which is one type of machine learning where the methodology, compute methodology there can learn a lot of nonlinear connections. And basically it's a lot of linear regression equations all pointing at each other that crudely look like a human brain layering. Maybe originally it looked like that, but not much anymore. But obviously deep learning is much better at learning things from pictures, especially retina pictures, as you I'm sure all know. But just to demystify this for a moment, you can learn about all 3 of these technologies with dummies books, okay? In fact, they're like $17 on Amazon Prime. These are just software tools that we can download to put into our software that we're writing. There's no wizard behind the curtain here, right? You can literally buy dummies books on this and get started. So in fact, there are many high school students that know about big data and AI machine learning, as we'll talk about too. But of course, it's already changing medicine, right? So you got blog posts that say it's changing medicine. You got Eric Topol writing his book, Deep Medicine. You have some journals that are so old, they're new again, like AI in medicine. You have fields like radiology, right? Radiology was a journal. Now there's radiology AI. That is a sub-journal for them, right? Imagine ophthalmology journals with an AI sub-journal, right? You have Nature Medicine, which was the most biological of journals, what I remember when I was training, and JAMA, which was the most clinical, both of which now have AI and data-driven calls for papers, and they're routinely publishing in this field. So obviously, even for us at academia, people are looking for these papers. And of course, we have products and services. So here are the first 3 deep learning tools or services approved by the FDA. You can see the back one there was only approved in 2017. So this is still a recent phenomenon, okay? And you can see the one in the back there was Arteris, which actually now has a UCSD connection. One of the founders is now at UCSD, looking at cardiac imaging, right, to evaluate cardiac strength and heart and perfusion. Viz AI is for stroke triage, so patients coming into the emergency department to triage for stroke right away. And then, of course, diabetic retinopathy. A lot of teams and groups working on diabetic retinopathy, retina pictures. IDX, I think, was the first company to get approval for their product or service. So I think Google beat them in terms of a paper, but IDX obviously has a product. These are the first 3. Here are the next 40, okay? So now this takes us to the end of 2019, actually the middle of 2019. You can see all the different fields of medicine on the right there. It's not just ophthalmology and radiology. It's going to be every field. And this just continues to exponentially grow. This is as of the middle of 2019. When I just looked yesterday for radiology, I now see more than 100 products and services with machine learning already approved just in radiology, okay? And about 70 of them approved last year during the pandemic, okay? This is the time to be innovating this. And to be super blunt to this audience here, it's basically going to be the case that if you don't end up inventing this, you're going to end up having to buy it at some point. There is no reason we in academia shouldn't be inventing these things, especially given the data we can organize together, okay? But if you don't invent it right now, you're going to end up buying someone's product at some point, because that's how fast and furiously these are coming out. All right, so this is an exciting time. But of course, AI in medicine goes way back, okay? Here are the original papers from the early 70s on AI and medicine. Ted Shortliff, obviously in his Stanford days, had this first paper, how artificial intelligence could guide clinicians to choose the right antibiotic, right? Of course, different bacteria have different resistance patterns in different hospitals. And so it was so amazing back then that they literally had the code of the software and the input and output in the paper itself on the left there. And on the right there, you can see early 70s, New England Journal of Medicine perspective, medicine and the computer, the promise and problems of change. We literally could have that title again, okay, in the New England Journal of Medicine. But what happened, right? In the 70s, then all of a sudden we hit the 1980s and the AI desert because there still wasn't a lot of data. It was hard to get rules out of doctors or out of any experts. And the field really floundered until it started to really resurrect in the past few years. Why is AI back in biomedicine? 4 big reasons. First, we have great hardware that we can use, computer hardware from the video gamers, because it turns out, as many of you know, the same video boards that we use with chips from NVIDIA and other companies, it turns out those chips are also great for really being efficient at deep learning. But we can buy them at consumer price levels, right? We don't have to pay millions of dollars. We're paying hundreds of dollars for these boards. In fact, it's so annoying sometimes gamers can't buy the boards because we're buying so many of them for our work, right? We have great software tools to use from great universities like Berkeley, from companies like Google that are so well used now that high school students know how to do it and use it. And there are a lot of online communities to learn from and to kind of post problems and get solutions. And then in biomedicine, we have enormous data sets to work with that are getting better and bigger. And there are 2 big ones. I'm going to talk, the talk is split into both of these 2. The molecular revolution, of course, DNA sequencing, but not just DNA, RNA, proteins. We're not just measuring more things, we're measuring them on more people and more cases, right? Not just DNA on a few, DNA on a whole bunch of people, right? So that molecular revolution is still exploding. But the other big data is electronic health record data. I'm going to spend a whole half of the talk just on EHR data. Again, another aspect that's so old, it's new again, EHRs. And then, of course, we still have unsolved problems that need answers. How do we predict what's going to happen next to a patient? Efficient drug discovery, population modeling for this cohort. What's the best thing to do for this cohort? And so many others. So the problems are there, but the data is there too. And that's why this field is just exploding here, right? Now, I'm going to start with the molecular world and how I got my start. And again, molecular revolutions exploit. I could be showing you a sequencer from Illumina with blinking lights. I could be showing a mass spectrometer looking at protein levels in the blood. I love showing these. If we were in person, I'd be taking one of these out of my pocket, these gene chips, because this is how I got my start way back as a postdoc in 1998. These things had just gotten invented. And still, to me, it's magical. You can take a sample, a biopsy, tumor from the OR, mouse model cell line, and you don't literally put it on the strip, but you extract out the RNA, and you got a readout of all the genes being expressed or transcribed simultaneously with these chips. But look what happened, right? Everyone started to use these chips in late 1990s, early 2000s. In fact, so much so that we said, you know, it's getting bothersome to measure them one by one. And on the right there, you see the 96 well plate version of this chip. And then there's even a 384 well plate version of this chip. And why the point here is that biomedical data and our ability to capture biomedical data grows exponentially the same way the computer world grows exponentially, right? Moore's law, computer power doubles every 18 months. Well, biomedical data is growing at that same kind of rate as well. Now, what happened in this field, right? Again, I could be showing you sequencers or other things, but I'm going to stick to this field for a second with RNA measurements. What happened in this field is everyone started to use these, right? This was the fellow and postdoc thing to do in the early 2000s. Go get a gene chip. Now we use RNA-seq, but go write some papers on what's different here and there. everyone started submitting papers, and the journals got overwhelmed with all these papers. They said, no more papers with these gene chips. That's it, okay? Unless you put your data in an international repository where the reviewers can double check the match. Probably important to do that. And maybe others can use these samples for something else. And that's what happened. In fact, the precedent for this concept goes back almost 60 years now to Genbank. Okay, most of you probably have never actually really used Genbank, but you might know it's a next door neighbor to PubMed, right? Same folks run PubMed and Genbank. And we put COVID sequences in there. A lot of folks use it for that. But the precedent has been set that if enough scientists use a molecular modality, you got to share the data at some point. Fast forward to August of 2012, where we hit 1 million samples just in this world of RNA. We went from 0 when I started to 1 million in August of 2012. Now, if you look in the data repositories, it's a European and a US version of these repositories, doesn't even make a difference. We're at 4.5 million samples, all available for you, okay? Because why do I say that? What do most scientists do when they get a big grant, right? They go make these measurements, they write a couple papers, and then what do they do next? They go get another big grant to go make more measurements. And meanwhile, the rest of these measurements just keep accumulating in these databases, sitting there waiting for you, right? If you don't want to get this data, no one's going to get to this data. All right? It's just sitting there waiting for someone to do something. And that's really what the story is all about. Let me give you some great data sets out there, right? These aren't opto-specific here, but I'll just give you ones that are well loved and used. Here's a great example, the Cancer Genome Atlas, okay? Sounds impressive. It is. 84,000 cases of cancer, including some UCSF ones in there, of 68 different kinds of cancer. And here it's not just RNA, but they got DNA, oncogene sequencing, protein measurements, antibody measurements, clinical data, which chemotherapeutic or biologic did they use here? Sequencing, they even have imaging data in here, right? They have some mammographies, they have some CT scans. And literally this data is given out to people, right? Probably the next target that needs to get hammered with a particular drug is sitting there in this data set, right? And just continues to grow, the TCGA. Now I run this next one, so I'm going to put a plug for import, immunology data, COVID data, transplant data, more than 300 different trials, including clinical trials data. We curate and give out at this website at import. So this is how, it's funny that not only does the data grow exponentially in these repositories, the number of repositories is also growing exponentially, okay? That's how much data we just give out to scientists like you, waiting for you to do something. Well, what are we trying to do with all this? We're trying to get to precision medicine. And what is precision medicine? Well, my definition for this is the customization of healthcare based on measurements we're going to get from patients. Of course, some are going to be molecular measurements like DNA and proteins and RNA, right? We just talked about some of those. But we might also get behavioral measurements, exercise measurements, even preferences, right? Patient can tolerate this side effect, really doesn't want that side effect. We're going to need to get a lot of measurements like that from patients. Maybe it's with molecules, maybe it's smartphones and apps, but we've got to customize medical care based on those measurements. But we can't actually just do it with those measurements. We need those kind of measurements on a lot of people. And then figure out actually what medical care worked on all these people so we can learn from them and apply it to this patient. And that's what's precision medicine. Now the phrase is 10 years old. The National Academy report came out 10 years ago of precision medicine. The link is here. And to me, it boils down to this one figure in the 100-page report. You can see the 3 ovals at the top. To get to precision medicine, we're going to need to get better diagnostics. We're going to need to get better treatments. And we're going to need to figure out our health outcomes. Is what we're doing in medicine really working? Or to be more clear, what's working, what's not working? And so the rest of this whole talk is basically coming up with better diagnostics, better treatments, better health outcomes, just using the data. That's the whole concept here. I'm going to go quickly through some of these here. All right, so diagnostics. What is a diagnostic? It helps us as physicians, as clinicians, to figure out if a patient has a disease. We might have imaging diagnostics, right? You might look at retina pictures, for example. I love blood tests. I love blood protein measurements because they're simple. If you can set up what's called an ELISA, enzyme-linked immunosorbent assay, 1970s technology, you can get these kits to work in a desert. You don't even need a refrigerator or anything. So my lab in the early days, we're trying to come up with new blood tests for disorders. Here's one disorder that we were working on called preeclampsia. Now, you might remember preeclampsia is when a pregnant woman has high blood pressure all of a sudden. It can lead to when it gets shooting high, it leads to eclampsia. The mother can die, the baby can die. But what happens is we have to take these babies out super early and make sure that they stay healthy as pediatricians. Now, preeclampsia, if you remember, the diagnostic for this is ancient. It's urine protein, not even a specific protein, just urine protein. So we looked at this disorder. We realized, can we come up with something better in the blood? Is there a blood test that we can make this more specific for preeclampsia? And so that's what we did. Linda Liu was a grad student involved. Bruce Ling helped us with proteomics. Matt Cooper got involved in a moment, you'll see. And what do? We literally type in preeclampsia into the databases, and we downloaded a whole bunch of databases, okay? So many people have been looking at preeclampsia from the protein side and the RNA side and DNA side. So we said, let's download everyone's databases and figure out what's in common, right? I don't care if that researcher saw something or this researcher saw something, but if they both saw something, then maybe there's something there. And so think about Venn diagrams, right? I'm going after what's in the middle there. So that's what we did here. And we turned the crank and we came up with this marker and 12 others. HPX is a marker called hemopexin. And you can see it's higher in the pink. That means it's higher in pregnant women with preeclampsia compared to the green, which were pregnant women without preeclampsia. And you can see a p-value. You can see we got some grant funding on the top left, March of Dimes. This is during my Stanford days, internal seed grant called Spark. We wrote a whole bunch of papers. You could see that at the link at the bottom right there. But what do you do next in Silicon Valley? You start a company on this. And this became a company called Carmenta. Now, let me be crystal clear to this academic audience here, right? I'm not showing you, Carmenta raised $2 million here. I'm not actually showing you the $2 million to brag about it. In fact, if you know, $2 million is not a lot of money to raise in Silicon Valley. But what I'm going to try to convince you is that $2 million is how the next scientific experiment is going to happen. We got these markers to work. We got the grants. We got the papers. The next thing that needs to happen is a prospective multi-center trial. But that scientific experiment can now be done in a startup company funded by private dollars. Or to put it a different way, do you know how hard it is to get a $2 million NIH grant today? The science continues in the startup companies. And that's why the introduction means so much to me, right? I tell my lab, if we want to change the world, we can't just keep writing papers about it. If we've actually believed we've invented something or discovered something, it's up to us. If no one licenses that, it's up to us to take it forward and develop the tests ourselves, start the company if we need to. In fact, I'm going to say it's our duty to do this. Yes, there are a lot of rules and regulations. I know them all, okay? But you can follow the rules and still be very transparent and actually make this happen. Now, I love this company, Carmenta, because you can see at the bottom right there, it's already been acquired. Now, some of you already know what that means, okay? You know what that means. So we went from downloading data off the internet, right, to doing the computations, to getting the candidate markers, to buying the ELISA kits, to testing samples in the freezer, to starting the company, to selling the company. Start to finish, 18 months. Inventors happy, investors happy. Of course, we can do more of these, and I'm giving away the secrets here because every one of you could take this kind of data and develop a diagnostic for a different disease, and we'd never step on each other's toes. That's how many diseases still need a diagnostic. Okay, I'm literally giving away the secrets. Everything I just told you how we did it is at the links here. Okay, I've tweeted about this many times. That's one diagnostic. All right, let's talk about another diagnostic, DNA sequencing. Now, hasn't really hit ophthalmology yet, not really yet, but DNA sequencing is taking off, okay, especially in medicine. I was still a trainee in Boston when the Whitehead Institute, you see the picture at the bottom left there, was sequencing the whole human genome, right? And even then it was miraculous that just a dozen or 2 dozen people could sequence the whole genome, right, in such an automated way. On the top right there is my hand literally holding a USB stick powered sequencer. This is an Oxford Nanopore mini iron. It's a no-brainer. If we have sequencers we're running off USB sticks, it's a no-brainer we're going to be using this method. Now, I'm not saying we're going to use this in every clinic or even every exam room, but at a certain point, we're just going to be sequencing more and more. For example, it's not just germline sequencing, it's COVID sequencing. We'll be sequencing influenza. We'll be sequencing, right? Because it's just there. This is taking off. Now, I got my start in sequencing in the late 2000s. I'm not a geneticist. I'm a data guy, as you know. But I got my start because of this patient here. Top left there. Now, everything's been talked about like crazy, so there's no hip by anything here. This is Steve Quake, who's a very unusual patient. In fact, he's a faculty member. He was at Stanford. And he starts sequencing companies on the side. So he's a very unusual patient. But one day, he's the red circle in the pedigree. One day, his nephew, the blue circle, did not wake up one day, okay? 19 years old, died sudden death. Okay, you can see sudden cardiac death. And so Steve Quake now, with his genome sequence already published, went to our good friend and colleague, cardiologist, Ewan Ashley, and said, well, doctor, am I gonna die of sudden death? And by the way, doctor, here's my genome. Can you imagine if someone came to your clinic and said, here's my genome, doctor? That's what this became in 2010 in Lancet. A team of almost 50 people wrote a paper. This probably was one of the most highly cited papers that year. And it became a prototype of how doctors and patients are going to talk about having a genome sequence. I'm not saying we did everything right, but this is the first patient to show up with a whole genome sequence here. Now, Ewan Ashley could give you an hour-long talk on the DNA, okay? And it turns out that he has no known mutations in genes that set you up for cardiac arrhythmias. Okay, that's good, okay? But that could be an hour-long talk. Russ Altman, our good friend and colleague, got involved because of pharmacogenomics, that for 150 drugs, we can say something about that drug. Is it going to work? Side effects, dosing, we can actually make some kind of call there. And then, okay, so heart was done, all the drugs were done. My lab was stuck with all the rest of medicine, okay, every other condition. And so how did we do this? Well, it turns out this paper came out in 2010. In 2008, I realized patients were going to show up someday with their genomes. I thought it was going to be 23andMe, and then all of a sudden Steve Quake came with this whole genome. And I realized we were not ready in the field of medicine, even with just a master list of all the SNPs, all the spelling differences in our DNA, and all the diseases, because we kept writing papers like this, right? Here's just one of so many genetics papers where they looked at some genes, they found some mutations, the mutations were associated with different diseases. And what we did is we said, okay, let's take 50 of these papers. I got a San Jose high school kid and said, over the summer, we want you to read these 50 papers and build a master list of and curate these 50 papers. And we wrote down every p-value and confidence interval and ethnicity and race, every detail we could for these papers. And we said, well, if a high school kid can do 50, how long and how many people would it take to just read the entire literature in genetics? And that's what we did. So we blew past 20,000 papers we manually read, hired a team in India to read all of these. Here are just 1,000 front pages of PDFs. I got a script that does this from the PDFs. But again, this is just 1,000 here. We did 20,000 of them. And in the end, we made a master list of every single gene, SNP, polymorphism, and every disease. In fact, I think NIH still believes we have the best list in the country. And so what do? Is we said, well, Steve Quake, we're not going to show you the mutations and the genes. We're going to show it to you this way, where here the font size, this is one of the figures from the papers, the font size goes along with what your risk is for the different diseases. And some diseases cause other diseases. So just because by genetics, you might not say have hypertension, you're not going to have hypertension, you are set up for obesity and type 2 diabetes. And if you get those two, then you might still end up with that condition. So it doesn't mean just because your genetics say you're set, there's the rest of the world too, environment. And then all around the edge were all the known published factors that we could mine the heck out of PubMed and give that as a prescription pad, let's say a prescription to Steve Quake. Well, Steve, these are things you should do to avoid these diseases. Like watch your smoking, exercise, diet, stress, alcohol. Yeah, probably don't need a genome for those 5, I admit it. But at the top there's pesticides. Well, Steve, you're at slightly higher risk for Parkinson's. There are 10 papers linking pesticides to Parkinson's. We didn't write the papers, we found them in PubMed. So if you're going to go in the garden, stay away from the pesticides. Well, I think this is how doctors and patients are going to talk about their genetics someday, right? With all these risks. And imagine coming back in 2 years and your font sizes are changed because you're not going to get that condition anymore, but you might get this one, right? So this became a way to think about genomes. Well, it's exploded over the last 10 years with non-invasive prenatal testing, cancer sequencing, neurological diseases, on and on and on. But what did we do in Silicon Valley 10 years ago? We started a company called Personalis. Came out of stealth mode maybe 8 years ago by doing the Million Veterans Program. So in fact, most of the Million Veterans Program, if you know that project at the VA, those genomes are getting sequenced by Personalis. Went through 3 rounds of funding, had our IPO a year and a half ago. And right now, I think if the stock market doesn't continue to collapse, the company is worth $1.5 billion. 4 faculty founders and 1 CEO as the founders. Literally started literally 10 years ago this week. Now, again, I'm not saying this to brag about $1.5 billion, but instead just to say, do you know how hard it is to get a $1.5 billion NIH grant? The science continues in the startup companies. That's the point. Why say the finish line is a paper for us when we can and should take it further? That's the whole point here. All right, let me show you a couple more examples and then get to Q&A here. Drugs, drug discovery. Oh my God, so much harder and so much more expensive. Matthew Herper is a great example. He writes this article every couple of years, at least he did when he was at Forbes. Just literally doing the simple math. How much are pharma companies spending on R&D divided by how many drugs are they getting out of their R&D? You know, and everyone thinks it's $1 billion per drug. Oh my God, that's an underestimate. Here are the top 12 companies back in 2013. And you can see it's costing the top 12 companies $3.6 and $11.7 billion per drug. Probably not sustainable. Now, I'm not here to pass judgment on the pharma industry or biotech industry. I love them too, okay? But can't we use all this data out there to figure out some drugs? Or maybe figure out new uses for old drugs, right? So that was the concept here. We didn't invent drug repositioning. It goes way, way, way back. Everyone knows Viagra and all the rest. But we said, can't we use all this data on diseases and drugs to kind of make a match here? And that's what we did. Turned this crank like crazy, dozens of papers published, so many that we're not doing any more of these, so many people in this field. Here's one of the most recent ones. Bin Chen did this when he was at UCSF. Now he's at Michigan State running his own lab. This is a condition called hepatocellular carcinoma, so liver cancer, nasty condition. 350,000 people a year get liver cancer, but very few in the United States. And I think, okay, call me skeptical, I think because of that, we basically have no pharma and biotech companies working on liver cancer. This is an India-China problem, right? Because of viruses and alcoholism and all the rest. So that's crazy. So many people in the world get this, so few companies working on this. Well, we turn our computer crank and this is what we get. We get a drug that we think could work. Now, what you're looking at here are pictures of mice livers infused with human liver cancer. And you can see the one on the left is control, so that's how nasty it can get. The one in the middle is a drug we predicted could work called nicolosamide. Now, nicolosamide we give for worms, right? GI tract worms. In fact, it's a veterinary drug too, to take care of the worms if you might have in your GI tract. Nicolosamide doesn't look much better there, but NEN is nicosamide ethylolamide, which is a particular salt of nicolosamide that lets the mice take it orally. Okay, so they don't even need to get injected by nicolosamide. I can see it looks a lot better there. Okay, first experiment, not bad. Here are the glowing cancers. Okay, it was glowing big and now it's less big on the right there. So that also works. And here we did the experiment treating the mice with the other existing drug, which isn't great, called sorafenib. At the top, you see no treatment. Second row is drug, that's new drug that we're predicting. It looks better. Sorafenib also looks better, but you put the two together and it's better than either one separately. So that means there's probably synergy here, right? So this drug might be working in a different way than sorafenib. So if we're going to run the clinical trial, then we're going to dose these patients with both drugs here, right? That's the concept here. And this drug, okay, right, look, remember, there are no companies working on this. If we don't do this in academia, nobody's going to do this, right? That's our responsibility here, right? We got so many other diseases that we can target this way, right? Probably the drugs are already sitting in the pharmacy or not. Maybe there are drugs that we think failed for something else, could go into something else here. But what did we do in Silicon Valley? We started a company called Numedi. They've raised millions of dollars, and now they're focused on a particular nasty condition called idiopathic pulmonary fibrosis, which is when the lungs start to fibrose. Two existing drugs out there, neither works really well. Patients don't like them. So obviously, I'm not saying we have the solution to all of drug discovery here. I'm just showing you how we can do it with just existing data through academia. And that's what we got to do. All right, so that's the molecular story here. Now in the last few minutes, let me talk about the other big data, electronic health record data. Like I said, so old it's new again. And boy, is it costly. Here are just 3 press releases on how much it costs to put in electronic health record systems. Sutter are our neighbors here in California. And you can see that they spent $1 billion on their electronic health record system. partners, which is Massachusetts General Hospital and Brigham and Women's Hospital in Boston, and their affiliates, spent $1.2 billion on their EHR, and Kaiser spent $4 billion on their EHR. And amazingly, they all bought the same system, Epic. It's actually what we use in UC. So the price tag ranges between $1 billion and $4 billion for this stuff. Now, of course, it's more than just the software, it's workflow, redesign, whatever. We're talking billions. And here's the narrative I want to make sure you all leave with, that given the hundreds of billions of dollars we've spent on this data, it will be a national tragedy if we don't use this data to improve the practice of medicine. Period. I call this the most expensive data in America. We pay doctors to type all this stuff in. If we don't use this data to improve the practice of medicine, it's got to be safely, responsibly, respectfully. If we don't use this data, what a national tragedy this will be, given how much we spent. That's the point here. And that's why I'm at UC and UCSF trying to put all our records and figure out what to do with them. And with that, let me just reintroduce, who are we in the University of California? We're massive, right? Some of you might remember these numbers. 10 campuses at University of California, 3 national labs. I think Lawrence Livermore has the #1 supercomputer in the country, right? That's a UC staffed site. We have 200,000 employees. We're one of the larger employers in the United States, 250,000 students per year, 20 health professional schools, including 6 medical schools, right? But we have 14 other schools, right? Nursing, pharmacy, dental, veterinary, public health schools. We train half the medical students and residents in California, $2 billion in NIH funding, but we make $13 billion a year seeing patients. 5,000 doctors get a paycheck from us every month, but 100,000 doctors write orders on our patients. 12,000 nurses. And of course, we're not just shabby institutions. UCSF and UCLA are in the US News and top 10. And of course, we wouldn't mention that metric if we weren't in the top 10. We have 5 NCI Comprehensive Cancer Centers, 5 Clinical Translational Science Awards. And then we got these secret superpowers at UC that are so secret, we forget we have them. One of them is IRB reliance. Did you know that if any one of our campuses approves the IRB for clinical study, the other campuses more or less automatically approve it? It's like hours to days, maybe a couple weeks, instead of weeks to months to years, okay? Because they already see one campus approve it. And centralized contracting, let's say a company like Genentech to Google wants to do something with the University of California. If it's signed off at the office of the president, all 10 campuses agree to the terms automatically. They don't have to get renegotiated, right? This is the power of UC. And now we got this umbrella across the health enterprise called UC Health, University of California Health. We have this because aspirationally, we're trying to build accountable care organizations for the entire University of California. Now, ACOs are kind of a new concept. It goes back to Obamacare. But the idea is that we should learn how to take on risk and get paid that way. The difference is for an ACO, you're paid per member per month. No matter what that patient needs, we're getting paid per member per month, the same amount of money. Now, it's hard to learn how to do this, to take on risk. So we announced 4 years ago, we were partnering with United Healthcare to learn how to take on risk. And aspirationally, you can see a 10-year aspiration, we will eventually have a single ACO for the whole state of California, at least the entire University of California. I know UCSF has ACOs and UCLA and UCSD, but we will also have one that really encompasses the whole state. And so that's the aspiration. Now, here's the problem. The minute you've made a business decision like that, you got to ask yourself, well, whose style of medicine are we going to treat or use, right? UCSF does it this way, UCLA does it that way. Which is the right UC way to do it? And all of a sudden, when you have that question, you realize we got to put all this data in one place to figure out what is the right UC way to take care of patients. And that's what we have. So you can see the 6 kind of boxes at the bottom there, pointing at the top. And now Riverside is tiny, guys, right? They're a brand new medical school. They have some clinics, a couple thousand patients. The other 5, San Diego, Irvine, LA, San Francisco, Davis, are massive, millions of patients here. And we were happy with every day, all this data comes up centrally every month, except during COVID now, every COVID piece of data comes every night, okay? 6 to 9 in the morning, we get every piece of COVID data. And some of you might have seen some of our tweets. Millions of people have seen our tweets now. Just every day we put out our COVID statistics. Here's the latest data. And I'm going to get to the point. All of this data is sitting there waiting for you, especially part of the audience here at the University of California. The number we love to start with, 15 million patients treated over the last 15 years. That's incredible, right? We love this round number. because it's 5% of the entire United States population we've treated. But of course, if you only count when we put in Epic, which was in early 2012, right? We have 7 or 8 instances of Epic now across UC. We've treated just shy of 7 million patients now since 2012, 200 million encounters, 500 million procedures, 750 million medications, my favorite. You can see all the rest, 2 billion blood tests and vital signs. I like to say we have everything from Tylenol to CAR T cells. Tylenol is probably the cheapest thing we can give to a patient, right? A pill of Tylenol, fraction of a penny, to CAR T cells, when we take out your immune cells, train them to fight cancer and put them back into you, right? Probably the most expensive thing we can do, right? So we're not just Kaiser, we're just primary care. We have primary care, but we also have the most tertiary quaternary care you can imagine. We get data from all sorts of other sources, OSHPD, California state data, text data, all the rest here, and constantly harmonizing all of this here. This is what it looks like here, the 6 million or so patients in this part of the country. You can see all of our different colors. Yeah, you see a blob at Las Vegas too, because if you're sick in Las Vegas, you come to UC. The Hawaiian Islands are covered with our colors as well. Race, ethnicity, gender, of course, we can do all of those things. We even have all the telehealth encounters. This is public data. We've been showing this to Regents. Hundreds of thousands of telehealth encounters every week still going on. Now, this doesn't really happen in ophthalmology or even radiology. This is all the rest of medicine here. But we've completed over 1 million telehealth encounters already in the University of California. Imagine training the AI on that. Well, we're not just imagining, we're trying to figure out how we're doing this, okay? How do we actually figure out what does the common telehealth encounter look like? Can we get AI to start to do some of this stuff? Now, look, the UC has benefited saving millions of dollars with this database, okay? Improving quality of care, it's a no-brainer. Decreasing our own self-funded plan, right? A lot of our employees get healthcare from us, like me and many others. If you think through all the incentive errors, we really want to make sure the care we deliver to our own employees is the topmost, most efficient care, okay? Because it's actually a cost center to us, not a revenue center. So we've done a lot of that kind of work. Central management of primary care patients. I'll just give you one example here. Here are all 42,000 patients we treat for type 2 diabetes in the entire University of California, right? On one dashboard, imagine that, right? 5 academic medical centers all working together like this. Doesn't happen anywhere else in the country. You can see 42,000 patients. This is a refresh as of December. I haven't updated this slide more recently. You can see the vast majority taken care of by a primary care doc, but some are followed by an endocrinologist. You can see the latest hemoglobin A1C in the middle there. You can see is it increasing or decreasing? A lot of people sitting around with COVID, so it's increasing. And all along the bottom there, you can see eye health, kidney health, blood pressure control. You can we compare it to IHA national benchmarks. And this is how now all the diabetes professionals across UC are sharing their best practices constantly, okay? This is how we're gonna all show each other how to deliver the best kind of care. This is what we're doing here. I'm gonna skip forward a little bit here and get to how you all can get to this kind of data here, right? All right, here we go. So why is this even more interesting? Because the FDA now, to me, it's not just an NIH thing, it's not just an NSF thing, but even the FDA is telling all of us, this is how you do it. Okay, these are 3 guidelines and frameworks that have just been published in the last 2 years. The bottom one there on the left there, you can see proposed regulatory framework. This is how they're going to they're telling us how they're going to approve AI and machine learning software as a medical device. The middle one there, real world evidence. What we used to call clinical data is now called real world evidence, okay? If we can get it extracted from electronic health record data, how can pharma companies, biotech companies use this data to get their drugs approved faster? Synthetic control arms, virtual clinical trials. I'll show you some of those in a second. And the Digital Health Innovation Action Plan means how do we literally write a prescription for an app? Now 20 FDA approved apps already that you can write a prescription for that payers are now starting to figure out how to cover. Why aren't we building more of these? 3 important guidelines released by FDA in just the last 2 years, all 3 in this digital data space. If you don't want to get into this field yourself, get your trainees into this field. This is where it's happening. Even the FDA is telling everyone, this is how they're going to approve these things. Now, Vivek, our partner, and I, Vivek's a fellow, now a faculty member at UCSF, wrote a paper in JCI, the link is at the bottom there, 21 uses we found for real world data. Of course, it's synthetic control arms, digital approvals, biosimilar development, which is important in eye health, studying obviously quality and practice of medicine, all the rest. So take a look at the paper there at this link. But we found 21 uses and probably 200 uses really out there. How are UC researchers going to get to this? You're going to learn with your campus first. So if you're at Irvine, you'll learn with Irvine data. So SF, you'll learn with SF data. All of us are using the same data model called OMOP. OMOP just means this is what the tables and the rows and columns look like. You learn from you get taught, you learn if you don't know anything from the CTSI programs on your campuses, the Clinical Translational Science Institutes. And then once you get all that stuff working with your campus's data, we at the UC Health at UCOP, the Office of President, are ready to help you scale that data, okay? So we can't teach you how to do it. You're gonna learn on your campus first, and then when you're ready, we'll help you just run that same query, working query, across all the UCs. So you bring it, you bring your data, you bring your scripts, we help you run it. You gotta sign away your firstborn child if you give this data away to people, right? The usual kind of thing. You can't download this data, but we'll help you run. So it's safe, respectful, regulated use. Now, at the top there, you see, we have a fully identifiable database, right? We're doing quality improvement, operational stuff. You saw zip codes being plotted there. And then we have this completely legally de-identified version of the same database. Okay? It's so legally de-identified that all UC Health IRB directors have already signed off on this. In fact, they signed off, it's called non-human subjects determined. In other words, you don't even need to talk to the IRB to run the script now. We've done all that homework for you already. Okay, so if you're happy with the one on the right, you don't even need to talk to IRB. You got to talk to your folks to learn how to do this first, but we're ready to help you scale. And let's say you need an exact date or a zip code, then that's what HIPAA calls a limited data set. It's in the middle, and we could help you get to those as well, okay? So we built the whole thing for operations, quality improvement for the CEOs, the health systems. So now that it's built, we built the research version of it, okay? Same database gets used for operations and research. That's why it's working here. That's why it's already paid for, okay? If you know what I mean. This is how you're gonna do it here. All right, so let me skip ahead to the end. I think I am running out of time here. Oh, this is just proof. This actually already lives in the Azure cloud using a tool called Databricks. So Databricks sounds really cool. It's actually a Berkeley spin-out company. They haven't IPO'd yet. I'm guessing they're going to at some point. And this is a simple SQL query, there's a little bit of code there, just to show you that an AI machine learning researcher in the University of California can actually build an AI tool to learn from, you can see the arrow, 165 million ambulatory visits with data already sitting in the cloud, waiting for you. All right, let me just thank a whole bunch of people here. Skip through all this. Whole bunch of collaborators at UC and UCSF and Stanford, my Stanford days, and NIH. Whole bunch of people, an incredible team across UC that makes all this data flow, including the tweets you see every day on COVID. You see the scatter across the campuses here. We have a very thin team at the Office of the President at UC Health. It's really, all of these folks are really on another campus, especially the folks at Irvine. Incredible folks at Irvine that we've been working with now for years. I have to thank UCSF, Priscilla, Mark Zuckerberg, and the Bakar Foundation for endowment support. And my lab has been blessed with 20 NIH grants from these. I think there are 11 institutes of NIH written here. The ones in red give me more money. The ones in orange give me less money, but I still love them. A whole bunch of disease foundations who give us money to work on their diseases. I have to always thank my admin and tech staff at the top right. We'd never get a paper grant or anything out the door without them. I thank my family and all the folks who have hired me in the past, and my mentor, Zach Cohen at Harvard, who's still a good friend and mentor for me. So let me just stop there, see if there's any questions. Thank you.
Thank you very much. This was truly wonderful. Let's go to questions. Josiah.
Dr. Butte, thank you so much again for your time. My name is Josiah. I'm one of Dr. Brown's postdoc fellows. As a fellow engineer and physician in training, I just want to say I really resonate with your idea of translating academic research into products people can use. With that said, I was curious. I have two questions. One of them is, what do you see are the biggest challenges to translating academic research into products? And then the second one is I'm wondering if you can comment on the tension, maybe there's no tension, between balancing your investors' interests with pursuing perhaps ideas that don't necessarily look like they're going to pan out, but may in the end very much will pan out.
Yeah, great questions. Okay, the first is really how do we do this? And so a lot of times we'll hear in academia, especially when you're an assistant professor, associate professor, how am I going to get tenure, right? In reality, a lot of people end up getting tenure, especially in medical schools, okay? And so people say, isn't this counter to getting tenure? What I'm going to argue is that if you're taking on significant projects, significant research projects, they're probably going to end up being great papers and can also be great companies. But sometimes I think we don't take on challenging enough problems because we're afraid of failing, and that's a legitimate fear there. So you got to kind of hedge your risk, right? You got to have some things that you know are going to work and succeed. Yeah, I'll get a paper out of this no matter what. But let me take on these high-risk projects to see this could actually change the world if it works. And now, look, again, I'm deliberately naive. Okay, I'm going to admit that, and I'm guilty of making everything seem easy. I also admit that. But to me, I see a continuum between academia and industry, right? People are always saying, you might hear a senior mentor say, oh, that's the dark side. They went to the evil side. But I see it much more as a continuum. Think about it this way, that our job now as an academic, when I look at my job as an academic, my job is I am an innovator, okay? My job is to innovate things. And maybe I'll get an idea of something that needs to be innovated that is so obviously going to help people right away that I'm going to start a company on that. And UC and all of our universities let us do this, okay? I would have never joined UCSF if I didn't compare every letter of the rule at UCSF compared to Stanford, okay? I know every letter of the laws. There are very subtle differences there, but not minor differences, okay? We're allowed to do that. We're allowed to spend a day a week or exactly 384 hours a year working on companies. A lot of us don't choose to do that, but we have that even at UC. We can do that. So sometimes I'm going to get an idea that's so obviously going to help people that I'm going to start a company and try to get funding that way. But sometimes I might have an idea that is so important, but it's going to need a long runway to get there, okay? But if it works, it's going to be just astounding. But it's going to take time to get there. And that I'm going to do and develop in academia. But think about it this way, that my wife taught me this, okay? In the end, writing a great grant is probably the same exercise as writing a great business plan. If you're not going to change the world in 3 years, you're probably neither going to get money from an investor, nor you're going to get money from Bethesda, from NIH, okay? What is the number one criteria they look at in study sector? I've started on study sector for 20 years, okay? Significance, significance, significance. Is this going to move the needle, they keep saying, right? If it's not going to move the needle, you're neither going to be able to start a company nor get a grant. So that's the point here. And so in the end, does it really make a difference? Because our job now is to figure out how do we propose our ideas and get money from a table full of people to give us money to execute our ideas. Does it make that much of a difference whether it's on Sand Hill Road in Palo Alto or at NIH in Bethesda? It's the same exercise. How do I get this team of people to give me money for my idea? So again, you can split hairs and look at all the differences. To me, it's amazing how similar it is to start up a new company and start up a new lab. If you're not going to try to even change the world in 3 years, it's going to be hard to get that next round of funding. Let's see if there's other questions. Maybe I answered it.
All right, let's move to Sally.
Thank you again.
Great talk. I'm Sally Baxter. I'm an assistant professor at UC San Diego. I do comprehensive ophthalmology and also informatics and data science and NLM fellowship at UCSD. I have some questions about the data warehouse. I had heard UCSF is including imaging data and wondering if this is going to expand to the rest of the UCs and whether that imaging data will include ophthalmology data like fundus photos and OCTs and so forth. And then my second question is about just looking at counts for the data. So I've worked with the NIH All of Us research program, which is a similar sort of like, multi-institutional OMOP-based data platform. And they do have a data browser where you can look in conditions and get a sense of the general counts before you go through the data use agreement and all the effort to actually extract the data. And I was wondering if there are plans for something similar for the UC-wide data warehouse.
Yeah, absolutely. Okay, so the first is, let me answer the second one. Yes, it already exists. If you go to analytics.uchealth.edu, you got to do this from a VPN or one of the UC Health sites. You'll see a whole bunch of pages that the public can't see. And one of those is a way to enter in the analytics platform and just do the counts. Now, the counts are basic. It's kind of like what we used to have with I2B2 with UC Rex, and we're just at least replacing that functionality there. But of course, the data is there centrally, so we can do more detailed counts right away. You know, how many patients got this after this? So sometimes that code works, but it helps us to have that code working on your campus first, and then we can run the same code there. So we can do basic stuff, but complex things are definitely possible there. So the data is there, and of course it's OMOP because we have such OMOP expertise across UC. That's why we chose OMOP, because we got Picori P scanner, Lucille Machado, we got all of us. All of those things require OMOP, and so that's why we realized OMOP is a great vendor-neutral standard for us to use. It's so, again, another thing that's so old it's new again, open source code and all that. So that's why we love OMOP instead of forever being stuck with Epic. Okay, so we take all of our data out of Epic, all of our instances, and put into OMOP every month, every 2 weeks now. Now, imaging, much harder, much harder. The entire University of California signed what's called a vendor-neutral archive contract, a VNA, and right now the contract is signed, but there's not a lot of data going in there. I think, I'm ready to be proven wrong. I'm not so sure. I think it's just UCLA and UCSF putting in some images. Okay, so they all talked, and it's radiology, not even pathology or ophthalmology. And it's not even all the radiology, it's just echoes and I think ultrasounds. So because it's very costly on those campuses to get the data out of their systems and put it in, it takes FTEs and stuff like that. So whereas the aspiration is there and we actually have a contract signed, I'm not sure we're gonna move ahead with that contract. We'll probably end up as a UC figuring out another way to do images. But what I'm gonna push back and say, there is nothing stopping your clinical community right now across UC from getting all the chairs to talk to each other and say, let us build this across UC, okay? If you did that, we would be there to help, okay? And that is what radiologists, right? All the radiology chairs talk every month. All the CTSA directors talk every other week, right? The more those clinical departments all work together, man, UC Health, UCOP is there, ready to help. But right now, that hasn't really shown up yet, at least as far as I know in ophthalmology. Again, I'm ready to be proven wrong. I see the CT surgeons doing this. I see high-risk OB folks doing this. So a lot of clinical groups do talk to each other regularly. I'm going to strongly encourage ophthalmology to do that as well. And then you come up with the need, and sometimes you got to come up with the dollars too to help. And we'll work out the technology with you all so that we all get those images together. But right now, to my understanding, there is no UC-wide repository for ophthalmology images.
Do any of the other specialties look at like federated approaches rather than doing a centralized repository?
Yeah, I mean, so federation is always there, okay? You can always do that. And like folks know, again, the reason we've progressed so far with UC-wide repository is because we've had federated data before, right? With UC Rex and I2B2. The challenge with federated is that the strength is at the weakest link. And so what was happening with UC Rex is that every day you'd get a different answer because one site would be offline or something. So that's why we moved to centralized. And because we need this data now for operations, it cannot be federated. So what I would say is, again, to be creative here for a second, business model creative, is there a reason why perhaps you would want to share ophthalmology images for operations or clinical reasons, not just research? And if you can get if you can invent or discover a clinical reason that you're going to want to share data, then all of a sudden it happens much faster than just the academics wanting it. I'll give you an example. The entire University of California now is signing on for a single PACS system. Okay, we're gonna have one contract for PACS. And why is that? Because the theory is we want UC radiologists to cross cover each other at night. Why on earth are we sending this business for Nighthawk to Australia, for example, right? We're losing money on that, right? Why can't we just cross cover? We're all licensed in the same state, right? Bingo, millions of dollars going to get saved. All of a sudden, we're going to have a contract for a single PACS. Is there some equivalent of that in ophthalmology? I don't know. It's what you guys do. But if you can figure that out, then you're going to immediately see the dollars to pay for all of this, right? Just a couple of those use cases pays for the whole damn database, okay? It doesn't cost that much.
All right, let's move to Ahmed. Ahmed, are you there? Zach.
Hi. That was a really interesting talk. I was wondering if I could maybe ask some more general questions, and I'm not sure, like, these are maybe difficult to answer, but we'll see. So is data security, or like, are data security measures able to keep up with this kind of great proliferation of data influx and movement of data across platforms? I don't know, maybe some of that is mitigated through the use of like cloud storage systems. And then
Yeah, great question.
Was,
go ahead.
Can I ask my second question?
Sure.
If you want to answer. And then kind of operating under this assumption that more data points and types of data, all consolidated, make a better AI or machine learning platform. Do you predict that companies will eventually vie for proprietary use of patient data points? Maybe there are kind of legal protections against this. I'm not sure. But
yeah, okay, so great questions. Okay, so the first on data security, obviously it's a moving target. There's cyber security risks everywhere. You all know all the rules and things that you're gonna have to constantly learn, so do we. We use best practices at UC Health, but we also hire our own security auditors to make sure and keep doing penetration testing every so often to make sure that we're not missing something. So it's a hard problem, right? I think. Yeah,
I imagine it's very difficult to kind of keep up with.
Yeah, so it's incredibly hard to keep up with, but that's our duty is to keep up with it, right? So we can hire the best people we can, and we're going to, and we keep we believe that we're going to stay on top of it. But no one puts that out there saying, "Come hack me," right? I mean, no one's willing to say that. No, obviously, I mean, this is still a hard world, a tough world to deal with. So we believe that we're there, but it's not just that security. I think security from our own users, right? Let's say, I'll give you a worst case scenario. Let's say you come and look at all this competitive information, not HIPAA, not PHI, but competitive information. Let's say you know that UC Irvine is going to build up this or is going to open up a new cancer center so-and-so, and then you go join a competitor like City of Hope or Kaiser, right? That is going to cost us millions too, and we have no security on that either, right? So there's the data side of it, like hackers coming in and extracting data. That's also our own trainees and all our own folks, the people, the softer side of it. So there are no solutions for that. Companies deal with that all the time. But at least from the technical side, legal side, for sure, we cover that constantly here. The other questions of proprietary nature. And so, yes, I definitely do see that companies that see business models here and value are getting private dollars to start to try to take this data. Now, you see, we don't want to, and we're going to really, our general guidelines, we prohibit exclusive arrangements for data, okay? We don't want to give data to a company and us say, we're not going to give this data to anyone else, right? I think that's an important point. There's no way we would want to sign on to something like that. But I can definitely see companies definitely want to take this on. And they're gonna get the data any which way they can. I want to just point out one point here. Give me a second, let me just share my screen again. And this is directly in your field. And this, I think, is more of a worry than anything. So here I'm just showing you the JAMA paper. I think this is the Google paper, right? This is Google, yes. And everyone knows this paper, deep learning, diabetic retinopathy, right? And let me just show you, right? So there was no real data set here. So what did Google do here? You gotta look at this because it's really impressive. Where is it here? Yeah, 54 US licensed ophthalmologists and ophthalmology senior residents were paid for by Google, right? Google said, hell, this data doesn't exist. Let's just go pay 50 docs to read 128,000 images, right? Wow. How much did that cost? I don't know, make up a number. How much do senior residents get? It can't be that much more than when I was a senior resident, right? But boom, now Google has the database, right? And meanwhile, we're all arguing, are we gonna share our retina images across UC, right? When we're all on the same team. And Google comes in and says, well, let me just hire 50 docs to read 100,000 images. That, if we don't get our act together, guys, this is what we're gonna be buying soon enough.
Thank you. So let's move on to Ahmed. Are you awake now?
Sorry, I'm having a problem connection, but my question was if we are doing it on like institute level, is there a search option we can add in Epic so that we can individualize data based on the institute?
Yeah, so there is individualization already in Epic. So some, for example, it's in every field, right? Not everyone uses Epic differently. It's the same way across UC. For example, at UCSF, we really go crazy with breast cancer, right? Center of Excellence and all that. So a breast cancer doc at UCSF is going to fill out a lot more detail in the template for breast cancer than, let's say, at Irvine or somewhere else. So those templates are already different. But I am not going to be the one to tell a doctor to document more in Epic, okay? Never. I'm never going to be that person because I already know how much docs hate Epic, right? And already spending too much time documenting Epic. So that's the trade-off. My job, though, as a data guy, is how much can I extract out from what you're already doing in Epic, right? That should be my responsibility, not to ask you to document more. That's a non-starter. So there's a yes there, but there's also a no to your answer.
All right. We had a wonderful conversation, so maybe it's time for final comments and questions from Dorota.
Oh, thank you. Thank you, Atul. What was wonderful, and to see the breadth of the research that can be made with data. It's big data. It's pretty amazing. I actually want to come back to ophthalmology as a field in general. I think some part of the difficulty is that it's a lot of is descriptive and it's not based on images. And I was wondering if there is a special push, or it could be, because I think it is worth to have a special push to normalize the information so it is better searchable. And second of all, I personally think that ophthalmology data is extremely valuable for neurodegenerative data in general, as we can have a data from ophthalmologists. Everybody has data from ophthalmologists in what they want in their lives, while we don't have the evaluation of our cognitive, for example, performance. And as we know, eye is the central nervous system, and so on, so on. So I think this data is much more valuable than only for ophthalmology. That's why I'm just kind of expecting there should be more push from many fields to get this data.
Absolutely. Look, researchers and academics have been wanting data harmonized and collected for a long time, right? And it predates me. But there's only a certain volume of voice an academic has in UC system or anywhere. And what I'm trying to encourage you to think about is if you can figure out clinical and operational reasons for this to happen, it will magically happen, okay? Our entire database, to get started, way back when I joined UC in 2015, we got launched about 2017. Our pilot to get all of our health system data together cost us only $1.25 million, okay? That's it. And by the end of $1.25 million, we paid 50 people across UC to get this to work. We had all the data in one place. We're not talking even 10 million or even $5 million here, right? To get started, it's pretty cheap because we had a business reason to do it, okay? Now I'm gonna leave you guys with one important point. Here's a killer clue, okay? If you want a good business reason to put all this data together, and I know it's not the images, but if you proposed one important thing that would get you the dollars to do this, it would be biosimilars. You all know the drugs you use. You all know the biologics you use. You know biosimilars are there. If you went in as a UC ophthalmology group saying, we want to pilot better use of biosimilars now, okay? You know the dollars would be there for you to put all of your data together. I'm just going to leave it at that. Okay.
That's a great suggestion.
Okay. If you were the first at UC to say we are going to go to 100% biosimilars, do you know how many resources you'd get right now from UCOP? Let me just leave it at that.
Thank you so much.
Hope this helps, guys.
Thank you. We will be in touch with you for sure in many different projects. So hopefully you're open for new collaborations.
Take care.